Okay. Good morning, everyone. Welcome to day 2 of our Epitranscriptomics symposium. We are excited to have everyone here, and especially the speakers for the lineup this morning. Thank you very much, everyone, for making time for this event. In the interest of time management, I think I need to be very brief with the introductions and just keep right to schedule so that everyone has as much time for the science and the questions. Our first speaker this morning is Dr. Richard Gregory, Chair of Pediatrics, Stem Cell Biology, and Professor of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. And without further ado, Dr. Gregory will allow you to just dive right into your presentation. Thank you so much for making time.
Yeah, thank you, Scott. Just going to share my slides. Well, I just wanted to start by thanking Sammy and Kate for organizing this interesting meeting and for the invitation today. What I'd like to do is talk about our work on 2 different modifications, M7G and M3C. I'm going to introduce the methods that we developed for mapping these modifications throughout the epitranscriptome, and then talk a little bit about our work where we've applied these methods to investigate the role of these modifications and the relevant methyltransferases in the context of mammalian stem cells and cancer. Okay, so since I'm the first speaker in this tRNA session, I thought I should highlight the relevance of tRNAs. Of course, we heard a lot about M6A and messenger RNAs yesterday, and that's certainly the predominant modification on a messenger RNA. But once you start working on different modifications, basically all roads led us to tRNA. There's at least 170 known different modifications. At least 50 of them are known to occur on tRNA. And an average tRNA molecule is only about 70 nucleotides long, but it contains 14 distinct modifications. Some of these can be simple in terms of a simple methyl group. Others are much more elaborate, and especially those that occur in anticodon, which often involve a cascade of different enzymes. So today I'm going to be talking about 2 modifications that occur on tRNA. One is M7G that occurs at position G46 within the variable loop of a tRNA. And the second is C32, which occurs in the anticodon loop. So we've worked for several years now on M7G, and in the interest of time, I'm basically going to summarize most of this work. I'll put a little bit more emphasis on the method itself, since that's one of the focuses of this symposium. But we started working on M7G several years ago, and we developed a method for mapping it. And then we looked in the context of embryonic stem cells and found that it's required for stem cell proliferation and differentiation. And then more recently, we identified a role for the enzyme METL1, which is responsible for M7G modification. It turns out to be a potent oncogene and is highly relevant for many different types of cancers. I'll talk a little bit about that in summary form later. And then most recently, we teamed up with structural biologists to be able to present here this is a cryo-M structure. We also have crystallography as well, but basically to show the methyltransferase complex, which comprises the catalytic subunit METL1, together with its essential cofactor WDR4, bound to a tRNA substrate. And this, of course, helps to, at the molecular level, understand exactly how this complex is able to specifically recognize and modify a subset of tRNAs. And then I'm going to move on to the second topic today, which is going to be M3C, which is there are at least 4 known M3C methyltransferases in the human genome. And I'm going to talk about our methods for mapping M3C and also unpublished work where we investigate the role of these relevant methyltransferases. So let's get back to M7G. It occurs at G46, as I mentioned, in the variable loop region. This modification is important to help stabilize the L-shaped tertiary structure of the tRNA. It forms the methylated guanosine, forms a base triple with nucleotides in the D loop, shown here. To help stabilize the functional conformation of a tRNA, much of this work was done by Eric Fezeki at University of Rochester, and this work was largely performed in yeast. And in the absence of this modification, these tRNAs are unstable and rapidly degraded. And so very little had been done in mammalian cells, so we developed two new methods. One is involving an M7G antibody, we refer to as methyl-rib-seq, and a second chemical-based modification detection method that is able to map M7G throughout the transcriptome at single nucleotide resolution. And as I alluded to earlier, we did this in the context of mouse embryonic stem cells. We revealed for the first time the tRNA methylome in mammals, and we identified that, unlike in yeast, where this modification is dispensable under normal growth conditions, it's specifically required for growth under elevated temperature. In a mouse, mammalian system, METL1 is essential for the rapid proliferation and normal cell cycle of ES cells. And it's also required for the normal differentiation, in particular into the ectodermal lineage germ layer. So this is the crystal structure, partial structure of the yeast complex, which is the orthologous to the METL1 WDR4 complex in mammals. So let's get to the method development here. So for the M7G methyl-rib, we used a commercially available M7G antibody that we carefully characterized to make sure it was specific. And then we performed methyl-rib-seq. So this involved isolating small RNAs using a column to isolate RNAs enriched for tRNAs less than 200 nucleotides. And then borrowing from Tao Pan's work, as well as Todd Lowe's work that developed methods for sequencing tRNA, we treated these RNAs with ALK-B, which is recombinant demethylase that removes most of the methyl marks. And without this step, as you can see here, this is just looking at sequences that map to tRNA genes. You can see that without ALK-B, there's basically the tRNAs are represented in pink here. There's almost no sequences that map to tRNAs unless you do this ALK-B treatment step. When you do that step, you get substantially more sequences that map to tRNA, at least 50% of the total reads map to tRNA genes. And then when we did the methyl-rib, you can see that there's an enrichment for tRNAs. Almost 80% of all the sequence reads now are coming from tRNA. And then, as you might expect, when we knock out the enzyme responsible for M7G, this is a METL1 knockout, you can see that now there's basically no reads again that map to tRNAs. So this is a nice validation that the sequencing method is working and the M7G antibody is suitable for methyl-rib. And then we could identify the subset of tRNAs out of those that are expressed in ES cells that contain this modification. And that's confirmed here by methyl-rib followed by northern blot. And you can see, so this is the input tracks in the wild type and knockout mouse embryonic stem cells. And then when we do the IP in the wild type cells, you can enrich for these M7G modified tRNAs. This tRNA is not modified and it's not pulled down in this experiment. And then as an extra specificity control, we did the same IP in the METL1 knockouts. And as expected, we do not see enrichment of these tRNAs. So the methyl-rib-seq was informative, but of course it doesn't tell us anything about which site is modified or the relative stoichiometry of these modifications. And so for that, we went back into the literature and we found some old chemistry that was described in the 1970s that involved chemical cleavage of M7G. And so this method basically works, you use sodium borohydride that reacts with a methylated guanosine that leaves a basic site in the RNA that can be cleaved subsequently by aniline treatment. And so this method works very well. This is initial experiments where we treated the small RNAs with the chemicals as described just now, and then we analyzed the cleavage products by northern blot. You can see here, again, there's a very specific cleavage fragment detected in this, the RNA that's treated with sodium borohydride and aniline. This band is absent in the METL1 knockout, as expected. And of course, just as a control, these are untreated RNA samples. So it's a very specific cleavage that is detectable by northern. And we reasoned that we could adapt this method for high-throughput sequencing to globally explore where M7G occurs throughout the transcriptome. That's this method we call TRAC-seq, so it's tRNA reduction and cleavage sequencing. And on the bottom here, there's some TRAC-seq examples that were performed in mouse ES cells initially. And you can see the first and third row are the wild type and METL1 knockout RNAs, just the total RNA samples. And these sequences map to these different tRNA genes that are labeled here. And then when we treat the cells with the sodium borohydride sodium borhydride and aniline, you can see there's a discrete drop-off of reads shown and highlighted by these red arrows that represents the guanosine that was specifically M7G modified. And again, as a control, you see that we do not see this drop-off of reads in the MEDAL1 knockout cells, again, as we would expect. So by combining and comparing these methods, and there was some discussion yesterday about the relative utility of methyl RIP for M6A detection, we would argue that this is a complementary extra, orthogonal assay for validation. We compared the tRNA subset that we identified by methyl RIP-seq, which was this 21 tRNAs that are modified, and this overlaps almost completely with the tRNAs that we identified by TRAC-seq. There's only one tRNA that we failed to identify by methyl RIP. So by these two orthogonal methods, we're able to define the tRNA M7G tRNA methylome in mouse CF cells. This modification occurs, as I mentioned, always at position 46 in the variable loop. It occurs in the context of this sequence motif, and this guanosine in the center is the modified G. And then again, as expected, based on the work that had been done in yeast, when you lose this modification, as shown here by northern blots, this is a MEDAL1, the wild type ES cells, MEDAL1 knockout in the second column. You see there's decreased abundance of these M7G tRNAs, and then this can be rescued by reintroduction of the wild type MEDAL1 cDNA, but not the catalytic mutant version. So this is all very consistent with what we would have expected. And, I'm not covering the work we did in the ES cells because that was published a few years ago, but I did want to kind of summarize and show a little bit of data about the role of MEDAL1 in cancer. This is something that we're particularly excited about. We identified MEDAL1 as a new oncogene. It's the gene encoding MEDAL1 is commonly amplified and overexpressed in many different cancer types. Shown here is immunohistochemistry for MEDAL1 in glioblastoma and adjacent normal brain tissue. And you can appreciate that in the normal brain, MEDAL1 is barely detectable, but is dramatically upregulated in glioblastomas. This elevated MEDAL1 expression correlates very well with poor patient survival. And indeed, if we knock down MEDAL1 in a variety of different cancer cell lines, but just shown here as a human glioblastoma cell, and then put these cells into mouse xenograft experiments, you can see that knocking down MEDAL1 dramatically suppresses tumor growth. And so therefore suggesting that MEDAL1 is important in the context of cancer. And then raising the possibility that maybe MEDAL1 itself could be oncogenic and can enable oncogenic transformation. So to test this, we overexpress MEDAL1 together with its cofactor WDR4 in non-transformed but immortalized mouse embryonic fibroblasts. And normally these cells cannot grow in soft agar, and this is an anchorage-independent soft agar growth assay. You can see when we introduce the wild-type MEDAL1, but not the catalytic mutant, this then allows these cells to grow quite dramatically and form these large and numerous colonies in soft agar, which is, of course, a hallmark of oncogenic transformation. And we put these cells into mice, and of course, they form tumors. So by these classical cancer assays, MEDAL1 is a bona fide novel oncogene. So to investigate the underlying molecular mechanism, we performed TRAC-seq on the control cells, anti-vector, just the normal MAFs, those that express either the wild-type MEDAL1, which are transformed, or the catalytic mutant MEDAL1, which are basically equivalent to the control cells. And what we noticed is that it was the same subset of 26 tRNAs that are modified under all of these conditions. So when we overexpress and have hyperactivated MEDAL1, we're not getting new substrates that wouldn't normally be, but rather we're getting increased M7G, which is shown here by a mass spec, on existing tRNAs. And so we interpret this to be that basically for any given tRNA, they're not necessarily stoichiometrically 100% modified. And therefore, when you have excess MEDAL1, you can have increased methylation on these normal substrates. And this is reflected both in the relative increased abundance of these tRNAs. They're shown here in red, the M7G modified. You can appreciate this is RNA sequencing, but there's a slight increase in the abundance of these M7G tRNAs compared to the control cells. And then furthermore, when we do this analysis, where we look at changes in cleavage score, which of course is reflective of the modification, and then plot that against change in abundance, we see that there's these two tRNAs that are kind of outliers. Well, first of all, there's a positive correlation between cleavage score and abundance, increased abundance. But the tRNAs that are most affected are these two, this methionine and this, what turns out to be particularly important, arginine TCT. And then this, therefore, allowed us to identify, and I'm not showing the ribose-seq that further supported this, but we suggested that maybe this tRNA itself could be oncogenic and is perhaps what's driving or downstream of the effect of MEDAL1. And so our collaborators in the UK, Kostas Salepis, they performed this experiment with human AML cell lines transplanted into mice. So these are AML leukemias. And you can see that the empty cells are shown on the top here. When they overexpress the wild-type MEDAL1, this certainly increases the burden of the cancer cells within these recipient mice. The catalytic mutant does not. And then this effect is largely phenocopied by overexpressing just this particular arginine TCT tRNA. And that's also reflected in shortened lifespan of the mice that receive either the wild-type MEDAL1 or the individual tRNA. So I'm just going to briefly summarize this part of the talk. What I've shown is that, and what we found is that in many different cancer types, you have elevated MEDAL1 expression and activity. This leads to increased modification of a subset of tRNAs, in particular this arginine TCT. This then leads to a remodeling of the translatome such that mRNAs that are enriched in this particular codon that requires arginine TCT, in other words, arginine AGA, these genes are enhanced. The production of the protein from these mRNAs is stimulated by having extra tRNA available for these mRNAs. And these mRNAs are not randomly distributed, or these codons are not randomly distributed throughout the transcriptome, but they're rather enriched in certain subsets of mRNAs that include cell cycle and growth promoting genes. And so this is the model that we proposed and recently published about the role of MEDAL1 in cancer. Okay, so I'm going to move on now to the second part of the talk. This is talking about M3C. There are 4 known methyltransferases in the human genome that are responsible for M3C. And we became interested in this several years ago from this study that was published in JBC. And it suggested that perhaps M3C may exist in messenger RNAs. And this is one of the reasons we became interested in this initially. But even in this study, you can appreciate that certainly if you look at this axis here, so certainly tRNAs are very heavily modified by M3C. And then if you see that this is 10 minus 10 to the 5, the relative amount of M3C in the mRNA is hugely less than what's detected in mRNA. And even if you look at the isolated mRNA itself, you can appreciate, as I mentioned, M6A is the most prevalent mark within mRNAs. And M3C, if present, is perhaps at least 1,000-fold less abundant than M6A. So this raised the possibility perhaps M3C is very low stoichiometry across the transcriptome. That's one possibility for these results. Another is that it's perhaps high stoichiometry, but it's only occurring on a few genes, or the mRNAs from a few genes. Or thirdly, perhaps this is some background from contaminating tRNAs. And so, of course, you need a method to be able to map M3C, to be able to address these 3 possibilities. So again, we went back to the literature and we realized that if you treat RNA with hydrazine under high salt conditions, this leads to a basic site that again can be cleaved by aniline treatment. And so we just showed this by Northern blot initially, and you can see that this cleavage is again very specific. You see that the cleavage band we anticipated is detected here. The other thing you can appreciate is that this tRNA is almost stoichiometrically modified. There's almost a complete cleavage and conversion of the full-length tRNA into this cleavage fragment. And this, as I mentioned, occurs at the position 32 of the tRNA. So we call this method HAC-seq, hydrazine aniline cleavage seq. And we applied this in an unbiased way to comprehensively define the M3C epitranscriptome in human cells. The results were slightly disappointing in that we identified all the known things and very few new things. But what we could identify is that M3C occurs in mitochondrial tRNA, these two, serine and threonine, at position 32, that was already known. It occurs at position 32 of serine. It occurs at position 32 of threonine and arginine. Again, this is known from studies in model organisms as well as some work in mammalians. And then it also occurs in a subset of tRNAs, this serine and this leucine at position 47 in the variable loop. And it occurs at position 20 in methionine. So we're able to show exactly where M3C is. We found no evidence of M3C anywhere else except in tRNAs. But nevertheless, with this method in hand, and we published this work a couple of years ago now, we could address what's the role of the individual enzymes in the human genome. And so in yeast, there's a single methyltransferase that's responsible for all M3C32. And you can see this by mass spec. This was identified by Tom Suzuki's lab as well as Eric Fuzecki's lab. And you can see from their studies, this was identified in 2011, but when they knock out the gene encoding TRIM140, you can see by mass spec, basically all of the M3C is gone. However, the situation is different. There are 4 known M3C methyltransferases in human, METL2A, METL2B, 6, and 8. And the role of these is largely undetermined. However, as we're working on this, 3 different studies came out that reported that METL8 is exclusively responsible for modifying mitochondrial tRNA. So we also had data that supported this. So we kind of stopped working on METL8 and we focused instead on METL2A, 2B, and 6. So what is the role of METL2A, 2B, and 6? So we generated human cell lines, HEC293 cells that are deleted for either METL2A and 2B together. These are almost, these paralogs are almost identical. So we knocked out 2A and 2B. We also knocked out individually METL6. And then in combination, we knocked out METL2A, 2B, and 6. And this is the results from the HAC-seq. The first column is the wild type cells without any deletion. The second column is the METL2, 2A, 2B knockout. This is the third column is the METL6 knockout. And then the fourth column is the combined METL2A, 2B, and 6. And what you can see is this breaks down into 3 clusters. So the bottom cluster here referred to as C. This shows that these methyltransferases play no role in these modifications. And that includes the mitochondrial tRNAs, as we would expect. The position C47D, again, is unaffected by METL2A, 2B, or METL6. Position C20 on methionine is also not modified by METL2A, 2B, or 6. So let's zoom in now on these top groups, A and B. So anyway, so in a nutshell, what I'm showing here is this. METL2A and 2B are responsible for arginine and threonine modification. We could also identify there's at least 3 isodecoders that require unknown enzyme that remains to be identified. We could show that METL6 is largely responsible for serine modification. However, this particular serine, GCT, there's redundancy between METL2A and 2B. It requires that it's modified by both enzymes. So that's just a summary of what I just mentioned. But importantly, we wanted to look at the effects on translation. So we performed RiboSeq in the triple knockout cells. And what you can see here is when we look at codon occupancy or changes in codon occupancy compared to control, that there's a differential effect on threonine and arginine compared to serine. And even the effect on serine tends to be codon specific. So HUs are more affected than the other serines. We could confirm this by reporter assays. And in the interest of time, we did gene ontology on those mRNAs with altered translation. And this is highly enriched in genes involved in cell cycle and DNA repair. And not surprisingly, these cells have a phenotype. They grow slower. They have defects in the cell cycle, and they're more sensitive to DNA damaging agents. So in summary, what I've shown today is that METL2A, 2B, and 6 are responsible for serine M32 modification, and this can influence in a codon-biased way the translation of mRNAs that are enriched in HUs that include cell cycle and DNA repair enzymes. So just to finish then, I'm going to acknowledge Esteban, who did most of the M7G work. He has his own lab in Dartmouth now. Gia did most of the M3C work. She's joined Amgen recently. And then Erdem did most of the mass spec. And thank you for your attention.
Fantastic. That was a really information-packed seminar. Thank you so much for sharing unpublished work. We have to move on and quickly, but there is a question here. Well, two questions, but one we'll have to save for the panel. Have you tested the hydrolysis patterns of tRNAs without HAC addition at 37 degrees C for the given times?
I don't think so, no.
Yeah, it was a question about his observation that we've noticed that tRNAs hydrolyze in single-stranded regions, specifically at the AC loop upon incubation with higher temperatures. This is a very specific question, and I think we can discuss this more at the panel. Thank you very much, Dr. Gregory, for your time and your science.
Thank you.
Okay, we move on now to Dr. Tao Pan, professor of biochemistry and molecular biology at the University of Chicago, who needs no further introduction.
Well,
this is great. Yep.
will it work?
It's working.
Yes.
Thank you.
All right. Well, thank you. Well, first, I would like to thank Sammy and Kate and Scott for inviting me to have the opportunity to discuss some of the work we're doing. So today we'll be talking about functional genomics and of course, RNA modification cells to microbiomes. Just my disclosure, I'm an SAB member of this TR therapy company. Okay, so this I normally show this sort of the gels that many of you have run yourself, and turn out that if you take the total RNA from a cell, you see a bunch of very dark bands, right? This will be an agarose gel, and this will be a acrylamide gel. And then, in fact, if you look at total RNA by weight, we throw some numbers around, but typically we say about 15 to 20% of those are tRNA. But if you really look at the copy numbers, there's, I would say, almost tRNA takes up about 75 to 80% of all cellular RNA molecules. And so again, we don't need to introduce any more about what tRNA does, but there's a bunch of other small RNAs, or they're sort of medium-sized RNAs, that meso-sized RNAs, if you wish, that we hardly study, which, for example, spliceosomal RNAs that are required for mRNA splicing, and SNO RNA for ribosomal RNA modification and other functions, and SRP, and the small ribosomal RNAs like 5S, 5.8S, and so on. So the goal of my lab, one goal is not to only study tRNA, but also integrate to have one sequencing library that reports all of these RNAs. Okay, back to the tRNA that, again, Richard had done a great introduction there, but I just want to reemphasize tRNA is extremely abundant. I would talk about somewhere around 10 to 100 million copies per human cell. The human genome has about 500 tRNA genes, depending how you count them, but the important thing is about 300 sequences, and any given cells, perhaps approximately 150, 200 of those are actually expressed. And there's high diversity, on average, many modifications, 13 modifications per molecule, and overall about over 40 different chemical types and so on. So one thing we're interested in also actually turned out there are 4 properties we really want to study simultaneously, and that is, I call them abundance, modification, charging, and fragmentation. So what does this mean here? Here, right? We have one tRNA abundance. I don't understand what that means. And each cell type of cell state, cells will generate certain kind of expression levels of tRNA that are needed to do recodons. And under normal conditions, the relative abundance of the isoacceptors or isodecoders themselves allows the cells to sort of do their supply and demand for decoding. But then charging is the tRNA, of course, carries amino acid. That when the tRNA carries amino acid, we call them a charged one, right? If it's not charged, that's also very stable in cells. So for any given tRNA species, there's a charging level measurements that should be done, for the reason being that actually charged tRNA level turn out to be very useful in response to stress or under adaptive conditions and so on. And modification is one of these things I normally call them a wild card that really is used for all the conditions. Either cells are happy going through proliferation or in response to stress and so on. And fragmentation, that is one of the tRNA fragments, right, is in the last 10 years or so, is it's been a hot topic of a study of biologically important group of bioactive smaller RNAs in the range of somewhere around 20 to 60 or so. Okay, so the goal here is to develop a method, now we call it multiplex tRNA sequencing, multiplex small RNA sequencing, that can actually measure all four properties of tRNA, and on top of that, also allows us to access the information about the other RNA families in the cell, such as snRNAs, no RNAs, and so on. So today, of course, this method we developed and published last year. I would introduce that to you today, and also very brief touch on our application of this to basic and clinical studies of microbiomes. I think I'm going to skip the clinical side, but I'm going to talk briefly about the application of basic studies of microbiomes. Okay, so back in, I would say 2015, 2017, this was our tRNA-seq version 1.0. We call those DMTRNA-seq, stand for demethylase tRNA-seq. So they originally, it was nicely done. So show, yes, use demethylase treatment, as you have seen in Richard's talk, that actually drastically increases the amount of reads in tRNA sequencing library. And the modification, what we found was in fact, things we used at the time are thermostable reverse transcriptase that reads through many of these RT interfering modifications efficiently, and leaving behind a mutation signature or sometimes a stop signature. And then charging is also the one we're able to develop a method for. So by 2017, we're able to measure abundance modification charging in one sequencing library. So you may ask, how do we measure these things? So for modification, the way we normally do this is through a thing we call mutation signature or deletion signature. Here's an example. There's actually an M3C modification is one of my favorite. Thank you, Richard, for introducing that. So this will be an example of a tRNA serine isodecoder sequencing through that. It has a modification as a position of 47D, or sometimes referred to as E2 position tRNA. So when we read through, when we do a sequencing library, we map our reads to the reference genome, where we find is this actually around this precise position of M3C, there's a mutation fraction, and here shown here is even 3 biological replicates superimposed on top of each other. Then we do for this particular site, we did a validation of primary extension, right, using a WinPRT that will stop at M3C. And it stops here, and that sort of reports a bit about, in fact, indeed, what we observe here from mutation fraction is consistent with this being M3C. And you can do this using mutation fractions for many of these modifications that have, they're modifying chemical groups at Watson-Crick phase, such as M1A, M1G, and et cetera. Okay, so how do we do charging? The charging, this is, again, one of the very old chemistry that using periodic oxidation beta elimination. So here, when we isolate the total iron from the cells, if you do this at pH 5, you keep the charged amino acid on it. And the periodic treatment will oxidize only the uncharged tRNAs, because periodic only react with the cis-diol. And cis-diol normally only exists in RNA at the 3' end. And then, so this treatment oxidizes the uncharged tRNA, but the charged tRNAs are protected because they have amino acid at the 2' or 3' end. Then after that, we do a beta elimination, just change the buffer and incubate a little bit more. At the same condition, we deacylate the tRNA to remove this amino acid. So this step removes this adenosine, the terminal adenosine that's oxidized. So leaving the uncharged tRNAs ending with CC, right? All tRNAs ends with CCA. So now the charged tRNAs from the cell before now will be read out in a sequencing library as A ending, whereas uncharged ones read out with C ending. So this allows us to measure a charged tRNA fraction. Okay, so then, but just sort of sometimes you get a bonus when you do something like periodic oxidation. Unknowingly to us, only a few years later, we found out, oh yeah, you can actually use the same chemistry to measure other RNA modifications. So here's an example. A QZ modification, right? This is the chemical structure of it. This is one of these known microbiome, gut microbiome derived metabolites that human cells take in to make their own modifications. So the Q modification itself has a cis-diol on the nucleobase, this 5-member ring on top of this. But periodic oxidizes it. So what we found was literally by accident is that if you use the right RT, we're using here the superscript 4. What happened then is that superscript 4 will read, here's a TR example, the histidine TR with this no modifications position, the WAB anticodon position. So if we treat without treatment of peridate, we read through this, we're looking at deletion fractions here. This actually is flat. It's not detectable. But once we treat the sample with peridate, then all of a sudden we are able to detect a deletion fraction. In this case, with histidine case, is a double deletion. And these are the cells we made. One cell, one sample. These samples come from cells that without any QZ modification. These other samples, all the TR is 100% modified. So this deletion fraction. And now you notice what's happening is that deletion fraction, of course, is not 100%. After all, even though we have 100%, we know the TR is 100% modified. It's shown by Northern blotting, but then even the deletion fraction for the histidine may be about 5%. So this comes, and then, however, even though there's a low fraction of deletion fraction, this allows us to quantitatively estimate or compare the Q modification fractions between two samples. So another bonus we got out of this is actually any TR modification with two sial position. This get also oxidized by peridate. This leaving behind a very nice mutation signature. And yet, so one lesson we learned from this story also is that when we measure TR modifications by mutation or deletion fractions, we actually not necessarily measuring their absolute modification fraction. Rather, we're sort of accessing them, if you wish. Perhaps we're getting these comparative modification information, right? So we can't tell from any single sample what the modification fractions are, but when there's a change in modification fraction under a different cell type or for a different cell type or under different cellular condition, we can see whether these modifications will increase or decrease, sometimes even quantitatively measuring the differences. So keep this in mind. So we do not measure the modification fraction per se, but we measure modification fraction differences. Okay, so that's being said, and then why weren't happy? We had our DMTRAC. Why weren't happy? So for many reasons we're unhappy, and even though there are many very very nice methods were published since 2015, and I think we stopped counting here probably around early 2022. So there's a thing about things I thought we could do better. So one is this lack of multiplexing. So normally we build sequencing libraries. We build one library at a time, right? We put one sample, 1, 2, and we go through all steps. And when we want to measure modifications, we have to do a lot of chemical treatments. If you want to do RP treatment with the enzyme, you have to add that to your sequencing library. The results may be highly variable and so on. So multiplexing means that if we were able to somehow barcode every sample very quickly and then combine many samples in the same tube, then all the downstream steps will be able to be carried out simultaneously all together. This allows us to increase the sample consistency and the robustness of a library building. And the other one is normally when we do this in basic science, we don't really do a lot of libraries, right? Normally we publish a paper with 24, 36 libraries. We're proud of ourselves. But if you do clinical studies, by the way, you will need hundreds of samples. And that is really, really hard to do when you, without multiplexing. So the other one is, it's a difficulty to work with, sample smaller amounts. This is easy to do, right? For something even for mRNAs, because people have worked this for many years, they were able to use very small amount of sample to do a lot of cool things. But for tRNA, it's still a challenge. And finally, something like when we started this, one of our interests was actually to study ocean water, which you can hardly have any material in there. So in any case, okay, let's rethink about this whole process. How do we do something to make all these things happen? And this is once we developed, it's called multiplex small RNA sequencing. This was published. I'm just going to give you some very salient points here. The key here is that here's our the key is we come up with this the innovation here is the hairpin oligo. This hairpin oligo has certain features that allows us to do all this stuff on the right. So here on the left. So here what we do, we take the cis here tRNA, but any total RNA. We're just looking at any RNA sample with 3 prime ends, and we do a first ligation. This is T4 RNA ligase. We'll ligate under a cool condition that makes things quantitatively. We ligate these oligos here. These oligos have a barcode in their hairpin loop, right? So we can have multiple barcodes. So each sample will be barcoded differently. Now we can combine multiple samples together. And then after that, we put these oligos have biotin on them. So we put them on a streptavidin bead. Now, when these things are on the bead, we can do our chemistry or enzyme treatment to do our commodification measurements. Give you a simple example. Some of you, if you have done something like CMC treatments for pseudouridine studies, that's an extremely harsh condition. You hardly recover. You lose, I don't know, majority of your sample. But by doing it this way, just as an example, a CMC treatment recovers 100% of all the RNA. Similarly, for even something like demethylase treatment, RB treatment, there was no loss of material. Then after that, we go through a bunch of steps to ensure that a lot of this, the hairpin loop has a sort of a unique feature. Is a 3' ribo nucleotide with a phosphate on them when it's chemically synthesized. This basically allows us to minimize the amount of the hairpin oligos, which generally is in the ligation reaction first is in excess over the RNA, right? So this allows us to really minimize any carryover final PCR product for the derived from the unligated free oligos. Okay, so we go through all this, do an RT reaction, and then after that we ligate a second adapter on them to the cDNA of these oligos, and the PCR can be done on bead. So once the PCR product is off the bead, so then you can now take that and pretty much sometimes you can just directly submit that for sequencing and so on. Okay, so one thing I want to mention now is that our method allows us to access information of tRNA fragments. So the very, very quick introduction, tRNA fragments, the small RNAs that regulate translation, cell-cell communications, and they are present typically around 1% of the level of the full-length tRNA without stress. When under stress, for some cells, they can reach up to 10% level and so on. And these small RNAs interact with many things, with RNA proteins and so on. Just imagine when the cell has up to 100 million tRNA molecules, even each molecule generate one fragment, we'll end up with like 1 million small RNAs in the cell. So there's a lot of tRNA fragments floating around. Okay, so how do we do this? This turned out to be in my MSRC library is a very simple bioinformatics trick. So for all tRNAs at least, our sequencing reads, right, for the full-length tRNA start from the 3' end. So if we do our mapping simply by bidding the reads where the 3' ends are, so let's say any 3' ends beyond position 60 of tRNA, we call them full-length. But anything below that, let's say here 50 to 59 will be roughly where the T loop is, 30 to 39 roughly where the anticodon loop is and so on. So we can count those reads, and then, but most importantly now, we can quantitatively compare the fraction of these reads to the full-length tRNA as represented here by 60 plus. So here's an example of a bunch of samples here. Each one of these are like a biological triplet kit here. So different cellular conditions. So we basically end up with about 1% level for these tRNA fragments altogether. So by the way, in those of you who are sophisticated, knowing, oh wow, once you stress with something, tRNA fragment level should increase dramatically. Actually, that works for HeLa cells and things like that. For Higgs cells, it does not. So just pay attention that your tRNA fragment level change under stress is actually cell type dependent. Okay, so here's another my favorite back to the M3C modification here. So what we're looking at here is M3C 47D modification in these two leucine isodecoders and all these serine isodecoders. So now we can look at actually the mutation rate of the full-length tRNA. Those are very high generally for the isodecoders. And we can look at the mutation rate of the M3C position for the fragment tRNAs. Those are the fragments with a cut in the T loop. So this is noticed very clearly. You can see that actually the modification level of M3C in the fragment is far less than the modification level of the full-length tRNA. This immediately indicates, yeah, M3C 47D actually matter for tRNA fragment biogenesis. Okay, so when we did a bunch of studies, I don't want to, just because now we have a tool, literally now we have a hammer, I just go around, look for nails, and we did a bunch of things. I'm just going to go through a very brief things of two of those. So one is the human cell stress response. So here's an idea: is that tRNA properties, of course, respond to stress, regulate translation, and translation of stress response genes under ultracellular conditions, right? So now we got to do, cells don't need to do translation, but they have to do something differently. Okay, so now, so here's how we did the experiment. We take our Higgs cells, we stress them in 3 different ways: heat, hydrogen peroxide, and sodium arsenide. this time we actually did polysome profiling. But the reason we didn't do RiboSeq, we did polysome profiling, is because we actually wanted to sequence tRNA in the polysome also, in addition to sequencing mRNA in the polysome. So that turned out to give us additional information we're interested in. Okay, so now we have basically for each sample, we have 4 conditions. We have total RNA, mRNA, and tRNA. We have polysome RNA, mRNA, and tRNA. Okay, and clearly these are the conditions that polysome profile shows the polysome levels went way down. So which is of course is what we expect. I just want to focus. We did a bunch of other analysis was in that paper, but I just want to focus once again back to the M3C modification. I think it's not only is Richard's favorite, but it's also my favorite. So anyway, there's these 2 modifications that he mentioned. The one is M3C32. That's in the anti-codon loop. That's present in these tRNAs. But there's also this other one, M3C47D in the E loop. That's in the variable loop of these type 2 tRNAs. But if you look at it, there's a cryo-EM structure with a serine tRNA in it. This 47D of modification is actually between the 2 subunits. It's between the 40S and the 60S subunits. So it's suggesting this modification will do something interesting, but very distinctly from the M3C32 modification, which will be, of course, the anti-codon loop is going to affect decoding a great deal. So here's what happened if we're just looking at, again, mutation rate changes among tRNA serine isodecoders. So under each condition, so this peak here corresponds to position 32, and this peak here corresponds to position 47. So now you can visually see that pretty much under most risk conditions, or not except under heat and arsenide, the M3C peak go up, and this is for total RNA, by the way, and the 47D peak go down. Hydrogen peroxide does the opposite; they both go up. This is just the control. So some of the tRNA isodecoders has a uridine at position 47D. Of course, there's a modification. So everything should be relatively flat, not necessarily because they also have M3C32. So this works out nicely. Here's what happened is just simply looking at the total RNA once again. When we do position 32, when we stress those things out, and the heat stress doesn't really change much, right? So these are the tRNA isodecoders with C at the position 47D, and these are with U at position 37D. So, but then some in arsenide stress and so on, this seems to increase. Somehow there's even under these stressful conditions, apparently cells keep adding M3C to their cognate tRNAs. But 47D goes in the opposite direction. So whether this is a degradation issue or this is a demethylase issue, we don't know yet. Okay, so then here comes the interesting things about is a polysome. What happened to this modifications on the polysome? So on a polysome, this is just an unstressed condition. You take your regular polysome, sequence it, tRNA-seq it, and then what happened is the polysome, we're looking at M3C32 modification for the tRNAs with a C47D or T47D. The polysome later compared to the input, I would say polysome collects tRNAs with M3C32 modified. So this immediately suggests that, wow, this M3C32 got to be important for translation because polysome loves it. M3C47D itself, this is the interface modification, right? It's not, there's a statistically significant increase, but it's sort of not as spectacular as the polysome one. It literally reaches almost 100%. Then if we do the stress conditions, by the way, then on the polysome, literally the M3C32 are already fully modified, right? You hardly get any change, maybe a little bit increase, but the 47D start to fall apart, especially under the arsenide conditions. So suggesting that perhaps under something like an arsenide condition where translation activity is way down, but and maybe the tRNA is helping to sort of reduce the interactions of the 40S and 60S interface and so on. Again, it just be consistent with this kind of interpretation. The other thing we wanted to do, was this was very recently. It was a single read tRNA-seq analysis. Remember, I told you, yeah, we're interested in this coordination of multiple tRNA properties, abundance, modification, sharding, fragmentation. Can we actually see the talk to each other at a single molecule level? Well, we don't really do single molecule Illumina sequencing, but we do sort of single read sequencing. Single read means we start out with one cDNA, coming from a single molecule, then of course on the Illumina chips get amplified millionfold, and we get to read information from those from that cluster. But nevertheless, I think we call them pseudo single molecule. That's close enough. Just take a look and see what we can learn from this. So we developed this computational pipeline called single read analysis crosstalk, or SLAC, and that you can get that free on GitHub. And that leverages the richness of TRC data and provides new insights here. I want to point out, I'm going to show you 2 slides about what we learned. One thing we learned from this.
Tao, I'm so sorry to interrupt. You're over already 2 minutes over time.
Okay. I'm going to skip the microbiome. That's fine.
Sorry.
I'm almost done with this. So then the correlation is even though I'm showing you this, I can't really tell you if you see a correlation that's causal or coincidence, right? Let's just keep this in mind. I'm not telling you every time we see a coordination, they're actually causal to each other. So here's my penultimate slide, I think, is to say this is what we're looking at here is a coordination of TRA M1A58 modification. This is a very common modification, the T loop of TRA to charging level. And I just want to point out that if you look at the iso acceptor families of TRAs that either have a wobble position of AOU or GSC, we get a very strong separation between those two. That meaning we have some kind of the charge to TRA has something, the charging, even at the charging level, I guess, has something to do with whether the wobble anticodon position is AU or GC. And turn out that if you look at the polysome profile, there's a very strong preference. The ribosomes love to read the GC codons over GC ending codons, a third codon position versus AU ending codons. And then this is my last slide. This is a little complex, but I just want you to think about this is that when you measure charging on a polysome, turn out that polysome, of course, the TR has an E site. E site TR is uncharged. So something like 1/3 of the TRA will be uncharged to begin with, right? This is reflected roughly around here. We look at the charging level differences between TRA and the polysome. It drops by about 1/3. And here we have, but there's something we didn't expect, that there's a M1A modification and the charging dependence. And this result simply means amount of time. Sorry, but just to say that this is consistent with if the TR is not M1A modified, it somehow doesn't like to leave the E side. It would leave the E side more slowly. I think that's the best interpretation of that. Okay, I'm going to skip out the microbiome, and just want to tell you that we have MSR-seq and the applications of major 14R properties simultaneously. You can do this with basic science or clinical science, and then access for a bunch of other information. This work was, the MSR-seq was developed by two former lab members, Chris Catensi and Chris Watkins, and with the help of another postdoc, Dr. Wen Zhan. And the SLAC pipeline was actually developed by a visiting graduate student from Luis Serrano's lab from the Center for Genomic Regulation, Barcelona, Spain, Javier. And these are the people who pay the bills, and thank you for your attention. Scott, I think you're muted.
Sorry. Thank you so much for your talk, Tao. Really appreciate the science, and I'm very sorry for putting on the accelerator.
No,
yeah.
Sorry,
yeah, we'll have time in the panel to get to more questions and details. So thank you so much for sticking around. Right now, I think without much time, I think we need to move on to the next speaker, who is Dr. Shenglong Zhang, from Associate Professor at New York Institute of Technology, and obviously a part of our RNA Center grant. Thank you, Shenglong, for your talk. Yep, you're up and running.
Slides.
Yep, you're up and running.
All right. So good morning or good afternoon, evening. My name is Shenglong Zhang. I'm an associate professor at New York Tech. I'm also the founder of this new bio startup. So today, I'm going to talk about the large-scale de novo direct and unbiased sequencing of RNA modifications using mass spectrometry. And this is a new platform we have been developing for direct sequencing and for quantitative mapping, not just one, but essentially all RNA modifications. I'd like to start with a very fundamental question. So do we now know the true sequence of RNA molecule? When we say true sequence, I mean the identity and the location of each and every nucleotide building block, modified or not. And this should include the unknown modification if there's one in the RNA molecule. We know very well from this two wonderful talk this morning, RNA are not only made of full nucleotides ACGO, they are made of many modifications. So far, more than 170 different modifications have been discovered. This number is still increasing. However, only a very small portion can be mapped. Most of them, more than 19% of those RNA modifications remain as dark matter stripes. And even for the method now, we used to map those RNA modifications. Typically, they favor one or two modifications at a time. There's no unbiased method that we can use to systematically sequence all modifications together in the RNA. So the question we're asking, is it possible for us to sequence all the modifications together so that we can know the true sequence of RNA molecule or even the entire transcriptome? We know it's not trivial to read the full nucleotides ACGO in the RNA. So how can we sequence all different types of modification together with this full nucleotides? So that's why we propose mass spectrometry-based sequencing. I know this is not commonly accepted, but we feel strongly this is probably one of the most promising methods that can be used to sequence all these RNA modifications. Because for sequencing, typically we need to have a signature tag to identify each nucleotide. So in terms of a tag, nothing is better than mass. Mass is a natural tag, both for canonical and modified nucleotides, and this includes unknown modifications. It would be difficult for large method now to identify all these RNA modifications. You may wondering how can mass be used to sequence all these RNA modifications? In fact, the conventional mass now even have difficulty to sequence of RNA. We know tandem mass spectrometry, also known as MS1, MS2, coupled with the LC, and they are considered as a golden standard for RNA modification analysis. So to use those methods, typically we have to purify the RNA and digest them into a single nucleoside. But they can't provide the location information, or they need a lot of method to provide this sequence information. So that's why we are developing what we call the next generation mass spectrometry-based sequencing. We would like to empower mass so that they can be used for de novo sequencing. We would like to develop this to a high throughput. And we also want to quantitative mapping those partially modified RNA modifications. So how can we de novo mass sequence of RNA? We know cellular sequencing has de novo sequencing in capacity. That's because of cellular sequencing is relying on the ladder generated by cellular dideoxy reaction. And this, of course, is a Nobel Prize work. So if we can generate a similar ladder for RNA, and then we can use mass spectrometry to de novo sequence RNA, the base calling can be made by comparing the mass differences between two adjacent ladder fragments and match this to the ones in database to base call each nucleotide for sequencing. One way to generate a ladder like this is to cut RNA once but a randomly amount of RNA, and this can be achieved by controlled formic acid. This will generate two sequence pool, a 5' ladder pool and a 3' ladder pool. Sequence can be can be read out from each pool, and they can reform for sequencing. And this is the typical data looks like. If we plot their mass against retention time, we can get this two Kinnear curve because the small fragments come out early with small retention time. The bigger fragments come out later with a bigger retention time. So they resemble like the sigmoidal curve. We can also differentiate the 5' ladder away from 3' ladder because the 5' ladder has a terminal phosphate, the 3' ladder does not have. So they're on the top of this two adjacent curve. So once we identify the ladder fragments, so we can de novo sequence of RNA, we can read it from 3' to 5' direction or 5' to 3' direction. The advantage of this method is that we can not only read the ACGO, we can also read the modifications. As you can see, those method exampled by those methylation. But this method also has a limitation. It's really because it's difficult to go high throughput because it requires pair end read to read the terminal nucleobase. So the terminal nucleobase are typically located in the small mass region. They're mixed with the small molecule, even buffer ion. So they need the input from another letter for this sequence information. So think about it. For one RNA, you need to have this two letters adjacent to each other for sequencing. If there are many RNA in the mixture, they're going to have lots of overlaps, making it difficult to sequence a mixed RNA. So how can we increase the throughput? Through many years of hard work, we now have different approaches, but for the interest of time, I'm mainly going to talk about this MASSIMM. MASSIMM is a computational data separation strategy. Remember, we rely on the formic acid to generate a letter for mass sequencing. If we look at the adding pair of this letter fragments that generated by this one-cut event, and their mass sum is constant, this equals the mass of the RNA plus the water molecule. So, for example, if you look at this 5' fragment 7 here, and the counterpart the 3' letter fragment 2, and their mass sum is constant. If you look at this 5' fragment 2 and their counterparts 3' letter fragment 7, they're also their mass sum is constant. Since each RNA has their unique mass, so we can computationally separate all the letter fragments and for sequencing each RNA in the mixture. Since this section is for tRNA, we have to talk about tRNA sequencing. tRNA is the difficult one to get sequenced by high throughput methods because the modification rich. It's also difficult for mass spectrometry to sequence tRNA because typically for mass sequencing of tRNA, it requires a highly purified single tRNA sequence, which is impossible to get. So this is one tRNA, a phenol tRNA from yeast. As you can see, this is definitely a mixture. tRNA has a mass about around 24,000 Dalton. So each dot here is indicated one tRNA. You can see this is a mixture, and this is the one for 76 mer with full-length CCA, but this is not the most abundant species. The most abundant tRNA is the 75 mer with the 1A missing at the 3' end. So despite the physical difficulty to separate them, with that unique mass, we can computationally separate them out and to sequence them like a single purified tRNA. So this strategy can reduce the need for physical separation, allow mass sequence any RNA, for example, a specific tRNA in a mixture directly. And this strategy could also help to increase the throughput of mass sequencing, allow sequencing potentially unlimited number allowed by the instrument. And this will, of course, help to pave the ways for large-scale global mass sequencing of biological RNA sample. One of the non-standing challenges for mass sequencing is how to differentiate the pseudouridine and the uridine because they have identical mass. So to differentiate the pseudouridine and the uridine, we treat this with CMC, like Tom mentioned before. And the CMC is going to selectively react with this pseudouridine and to form this complex. The mass is going to increase to 252. So after the acid degradation of this nether, and then mass is going to shift to the bigger mass region. And also this CMC is more hydrophobic. So after the acid degradation, the nether is going to move to more a bigger retention time region. And also this reaction is not 100% yield. We are going to see two letters. One is corresponding to a converted one is corresponding to a non-converted one. And this is all starting from the pseudouridine. And this is our sequencing data. You can see that can help us to pinpoint exactly where pseudouridine are located. And if there are more than one pseudouridine, we can see multiple branches. So this this ladder branch is kind of signature feature for partial single nucleotide substitution. And this can be used for partial RNA modification identification and to indicate, well, there's two different nucleotides, no matter they are modified or not, coexist in the same position. So how can we first identify those partially RNA modifications specifically? And this is our workflow. So once we have RNA, we divide it into these two portions. One goes directly to LC mass, and this could help us to reserve the sample diversity and modification information. The other one, of course, goes through acid degradation to generate the letters. If we see a partial modification, say methylation, at the intact level, we should see two intact mass with mass differences of 14,000. And this can be further pinpointed to be exactly where this modification, methylation occur by looking at the branch feature after the acid degradation. If there's a ladder two editing like AC editing transition, and then we should see a ladder two intact mass, and the mass difference should be 16. And we can also should see ladder two branches at the ladder fragment level. So this will help us to identify where are those modifications. Then we actually see when we sequence tRNA glue from a mouse leaf sample, we can observe this branch. So with this branch feature, we can know the identity. In this case, is a methylate A. We also know the case location is 58 position. And we also ask, can we further to know their stoichiometry? So how can we further quantitative mapping those partially modified RNA modifications? Remember here, the letter fragments we use to see for sequencing also carries relative abundance information. So for example, here, this two nucleotides, we can extract their ion current and integrate them and use the error ratio to calculate the stoichiometry. And this is also very common practice for the LC quantification. So I have to say, this is not an absolute quantification. This is just relatively percentage of stoichiometry of modified one versus modified at the same size, because they are coexist. They can use as an internal reference for each other. We also further verify this method actually is an accurate method. And this is one of the mixture is synthetic RNA. We know the ratio to begin with, and then we can throw that into the LC mass to measure the EIC. And we can also do some chemical reactions like about enable them and to measure the EIC. So you can see here, and that's very much after, if we measure the ELC, they are matched very well with the original ratio. We also look at a lot of different ratios. They have a little bit off, but it's all within the 5% error range. So indicate this is a relatively accurate method. Remember, we use the formic acid to generate a ladder. So what if there's acid nucleotides? If there's acid nucleotides, we should immediately notice that there's a mass change. If we look at the intact mass before and after the acid degradation. So this is the data when we sequence yeast phenol and tRNA. You can see they have different isoforms, but after the acid treatment, the mass decreased. This 358. This is really because there's a depuration in this acid condition. So we further pinpoint exactly where this is in this phenol and tRNA is in the 37 position. So with all those development, we start to sequence more tRNA. And this is one of the tRNA gene prepared from Professor Chen Qi lab when he was in UC Riverside. You can see we can not only read the for nucleotides ACGU, we can also read all those different modifications simultaneously. And not among this 10 modification sites, not all of them are modified 100%. We further determine the stoichiometry. To our knowledge, this is a lot of methods now can have this capacity to review this detail of information, and of course help to review the tRNA sequence. And this is the first time we directly sequence this tRNA at a single base resolution. And we know there are different types of isomeric methylation. If the methylation is in 2O position, and we should be able to differentiate them, because if there's a 2O methyl, and they're going to block the acid hydrolysis chemistry we use to generate the lead. So therefore, we should be able to see a gap, and the mass value should indicate the identity of this 2O methylated nucleotides. But there, of course, there are so many lot of different methylated nucleotides, and this is a little bit challenging. We have to review them case by case. Here, I just use one example. We take advantage of this specificity of the RKB enzyme. I think Tao mentioned, and also Richard mentioned before. So they have specificity for different nucleotides, like for M1A. Before the acid, before this RKB treatment, we only see one that indicates this 100% they are MA modified. But after this RKB treatment, this methylated lead is almost completely disappeared, and this converted to regular A lead. So we would like to say this probably is M1A, not M6A. Similarly, we can also tell this is M1G at the position 9 of this guanine. So this capacity actually is very important, because they allow us to track the change before and after the RKB treatment. And this could further address the big challenge we now have been discussing, could allow us to monitor RNA modification dynamics, to know first if they are dynamic or how dynamic they are. And also, of course, we can help to map the modification that have altered stoichiometry in different cellular or disease conditions. So the sample I have been talking about are typically a highly purified tRNA, not necessarily single tRNA sequence, but relatively easy to sequence. But I also would like to say we can also handle total tRNA, which is much more complicated. Say this is the data we collected from total tRNA sample, and you can see that different dots, each dot indicates a tRNA. And some of them matched with the one calculated from database, so they are showing in orange. Some of them can't match with the one calculated from database. That's mainly because of the tRNA has a partial modification. For example, we have you can see here that they have different intact mass. Their mass differences is like 14. That basically indicates that's a partial methylation there. And you can see that many not partial methylation. We can also detect the 3' truncation. Interestingly, in this sample, the most abundant 3' tail is not end at CCA, but CC. This, as you can see, you can see this is the most abundant tRNA in this total tRNA sample. But the most abundant species of this with the RNA missing from full-length CCA tail. This observation does not really reflect that's inheritable to the tRNA in the cell, but more likely be the result of some preparation, because we also detect the full-length CCA when we sequence mouse tRNA from extracted from mouse leaf, as I showed you before. But nevertheless, we have this capacity to observe those terminal differences tRNA. More interestingly, I think this Tao also mentioned about, we can deductively determine determine the charged tRNA. You can see they have 3 amino acid charged tRNA. Their mass differences matched very well. So this provides strong evidence they do have this type of tRNA in the total tRNA sample.
Long, I just wanted to tell you you're at time.
All right, this is going to be a last slide. Thank you, Richard. So if we combine our acid equivalent data, we would like we should be able to cite specifically and quantitative mapping tRNA modification. You can see this is the acid equivalent data. Ideally, we would like to de novo sequence each tRNA type in the tRNA. But this, of course, is very complicated data. We're still developing algorithm. But our current algorithm can not read the completed sequence of each tRNA type. We can read part of the tRNA type. So with this de novo information, we can search the database to find which sequence this tRNA, this part of the tRNA sequence belongs to. And with that information, and then we can find all the net fragments in the acid equivalent data. So one by one, we can get all the tRNA type and their sequence, and we can get the information about the identity, location, and the stoichiometry. So I'm running out of time. I would like to say this is just the beginning. There's so many things we can do, but with this, I would like to thank the people in my group who did this work. I would like to thank the collaborators provide the sample for those study. I also thank NIH for the funding and support. Thank you guys for your attention.
Thank you very much. There are no questions in the open dialogue right now. In the interest of time, as we're already about 5 minutes over, I would suggest that people save their questions for the panel. And Shenglong, thank you for joining us there in the next segment. I think now I would like to introduce the next speaker, who is Dr. Peter Dedon, the Singapore Professor of Biological Engineering at the Massachusetts Institute of Technology. And thank you so much, Peter, for making time for us today.
Well, thank you very much. And it's a privilege to be here. I want to thank Sammy and Kate for the invitation. I also want to thank Shenglong for giving my talk, and we can move on to questions right now. No, seriously, I've got a complementary approach to Shenglong's that provides some different information. First thing to do is to be transparent in terms of the practical applications of the science that we're doing. Second thing is to do proper attribution. I am privileged to work with an absolutely fantastic group of students at MIT and in a longstanding program in Singapore. And I'll tell you that I am just a few days back from Singapore, so everything you don't like about my talk is due to jet lag, not me. I don't have to really tell this story here. This is the Epitranscriptome Symposium, all the different modifications, all the different RNAs. My lab is basically interested in both the biology and tool building to solve the biology. We've come up with a bunch of different methods, heavily focused on mass spectrometry tools, next generation sequencing tools, but now also a lot of genomics and informatics tools that we're getting ready to publish with our collaborator, Tom Bagley. Today, I want to focus on mapping modifications in RNA. We've had some fantastic talks on nanopore sequencing of mapping of M6A and other modifications, showing the complex links to normal cell function and disease. I think that tRNA, as Shenglong and others have already mentioned, poses some unique challenges when it comes to mapping modifications and doing this quantitatively. Humans have over 250 expressed tRNAs, each of them containing 8 to 10 modifications. There's huge information content in that pool, highly dynamic, as you've seen so far. And as Richard and a number of people have pointed out, very clear, strong mechanistic links to disease. So it's very important now to figure out ways to map the modifications quantitatively on the individual tRNAs. So Tom Bagley and I have been working over the last decade to develop an omics toolbox, if you will, to analyze the tRNA epitranscriptome at multiple levels. And we've come to a model where reprogramming of the tRNAs and the modifications drives codon bias translation of response proteins. This is a model that's backed up by multi-omics that we've developed, mass spec, sequencing, all these different tools. Now, in terms of LCMS analysis of individual modifications, that's pretty straightforward to do. A lot of people are doing that. Here in this example, we can measure all the modifications in mycobacteria in one squirt. The patterns that you get in terms of relative changes are about 90% predictive of the phenotype. The heat map on the left shows these patterns that are very predictive of the time after hypoxia, but there's no tRNA sequence information. We developed a next-gen sequencing technology to quantify the copy numbers of fragments and tRNAs and all the small RNAs. But this gives you great information on absolute levels of different tRNAs. It tells you nothing, or very little, I should say, about modifications, quantifications of modifications in specific sequences. So how do you map the modifications in RNA? And we've heard some wonderful stories now about nanopore and single molecule real-time sequencing. I want to talk today about some bottom-up mass spec that complements what Shenglong just told about. Part of the motivation for this came from a recent NIH workshops and the National Academy workshops that were derived from that, really to ask the question, what are the best tools for modification mapping? And there were questions raised the other day about creating reference standards of modified RNAs, new synthesis tools to manufacture synthetic oligos containing site-specific modifications. And one of the most unappreciated things that was discussed the other day was sample preparation. That is such a monster. Vivian Chung and Pat Limbach, Mark Helm and I are working on standardizing sample preparation, shipping, and modification analysis by LCMS to harmonize all of the analyses that are being done. Okay, so what's the perfect modification mapping tool? It's got to be chemically specific, quantitative, both precise and accurate quantification. It's got to be sensitive, fast, high resolution, and cheap. Okay, nanopore sequencing checks the box on quite a few things here: sensitive, fast, high resolution, and cheap. It's not chemically specific in the sense of knowing the identity of the modification unless you have a priori knowledge with that organism. And even then, the quantitation is a bit fuzzy, but it's a powerful technique in terms of the throughput and this coverage. Mass spectrometry, as you've heard from Shenglong already, has some unique qualities that are to take advantage of. The chemical specificity and the quantitative precision are very good, plus the resolution. Now, right now, we face some problems of sensitivity, speed, and cost, and we're all working toward making this a far more facile and affordable approach. Now, what I'm going to talk about today is a parallel to proteomics, where essentially this panel on the left is a proteomics workflow where you isolate the proteins, fragment them with a protease, label them with an isobaric tag, mix all the samples together, resolve everything on the mass liquid chromatography feeding into the mass spec, do the fragmentation analysis that Shenglong talked about, and then mine the data with software equivalent to proteomics software. I don't need to tell you too much here because Shenglong already talked a lot about this. With mass spec, you pick the most abundant fragments, put them into the fragmentation chamber, break them into pieces, send the pieces to the detector for analysis. We can also deconvolute the charge envelope, which you get multiple charges for a big fragment that's negatively charged, and get an exact mass. You put all this together, and you can puzzle out the sequence of the fragment at hand, and then also the modifications that are present. And I want to show you now some examples of how this works. But first, there are problems here. Shenglong has pointed out some problems. Things like data-dependent acquisition are fuzzy. They're poorly quantitative because you're selecting things at specific times and you're sampling. What happens if you sample a big peak at the beginning or the end of the peak and you miss the middle? Well, so you can't just simply squirt a sample in and then come back and squirt another sample in and expect to get quantitative precision and accuracy. This is where isobaric mass tags, just like proteomics, come into play. The concept here is that you have a molecule that you link on the end of your RNA or your protein. The molecule's got two components. It's got a reporter and a balancer, and you use isotopes like carbon-13 and 15 to make different compositions of the reporter and balancer. When you put this molecule in the mass spec, at the fragmentation energy, the reporter pops off and gives you a signal. And now that signal has a different mass depending on the isotopic composition. And so each sample has a different label. We can get the relative quantity of that fragment in that sample by doing these isobaric tags. So the whole workflow looks something like this for RNA. You fragment the RNA. Now, Shenglong did it with alkaline nicking. We're going to use ribonucleases, and that has some limitations that we'll talk about. You label each of these with an isobaric tag that we've invented, mix them all together, put them in the mass spec, and now in your first quadrupole, the ion you pick may have a bunch of different tags on it, but they're all exactly the same M over Z value. And so now you send that fragment to the fragmentation chamber, break it into pieces. The reporters pop off, and that's what the little bar graph here is. You also then get the fragmentation to give you the sequence, and now you can get the relative quantities of that fragment with different modifications in it. So here are the challenges now to that whole workflow. You've got to do the RNA isolation, which is you've got to be very high quality. The fragmentation does present issues. New England Biolabs has just come out with a whole series of sequence-specific ribonucleases that we're now experimenting with. We're going to talk about ribonuclease T1 today. The tag labeling is a problem, and we've had to invent new tags that are RNA-specific. Chromatography of oligos is also a problem. You do not want to put ion-pairing agents in a mass spectrometer unless you want to dedicate that instrument to permanently measuring ion-pairing agents. Finally, the data analysis, as Shenglong alluded to, we need some custom software to be able to deconvolute all of this. So here's a problem. In the mass spec, DNA and RNA are actually very delicate relative to proteins. Proteins require a high energy to fragment them into pieces for proteomics. Much lower energies are needed for DNA and RNA. So the tags, the TMT from Thermo and the iTrack isobaric tags are structured, are designed to fragment at the higher energy of protein fragmentation. So we've invented a new RNA isobaric tag. Junzo Wu in the lab has now got this being piloted and tested. Today, I want to talk about applying iTrack tags as a proof of concept. It's not perfect, but it gets the message across pretty clearly. Now, here's some interesting problems. You put these tags on, we'll ligate them using a novel ligation technology to the end of the RNA. TMT tags, even though they're exactly the same molecular weight, elute differently because the isotopes cause structural differences, secondary structure differences in the RNA. It doesn't do that much with proteins, but in RNA fragments, the tags elute differently on the HPLC. That's a problem. iTrack behaves a little better, much more consistent elution on the HPLC into the mass spec, so we'll use that here. And now we're going to pilot this now with an 80-mer synthetic oligo, just to test out the system. The oligo shown at the top, if we digest with ribonuclease T1, which cuts at the 3' end of a G, we're going to get a whole bunch of cleavage sites in here. And in fact, this is the downside of using ribonucleases because a lot of the sequence is trash. We can't measure it. It's just dinucleotides or sequences that are too short to match to uniquely to any RNA. But here we're able to get 5 useful fragments out of this. So we can do our ribonuclease digestion. Now we're going to break that sample up into 6 tubes just to demonstrate precision, put our iTrack label on, mix these samples together, pop them in the mass spec, and down at the bottom you see our reporter, an icon for the reporter tag, and then the sequence. So when all is said and done, and we quantify this 80-mer oligo, we get very good precision based on quantifying any of the 5 fragments. We're up to plus or minus 5 to 10% coefficient of variation, which is actually very, very good for mass spec measurements. All right, so now let's apply this to a complex mixture of tRNAs. And here we're resorting to bacterial tRNAs because the pool is simple. It's just about 45 to 50 isoacceptors, so it makes analysis a bit easier. We're also taking advantage of a large library of gene knockout library of the Pseudomonas aeruginosa pathogen. Talented postdoc Jingjing Sun in the lab has analyzed all 5,800 strains for all their modifications. It's a beautiful story that we'll tell you about. She developed a high throughput RNA isolation system and then a high throughput mass spec system. A very brilliant undergrad at MIT came up with software now to deconvolute all the spectra. And now we have 50 modifications quantified in 5,800 gene knockout strains to get the network analysis done. Well, what we're going to do here is just pick 6 of those strains and look at the effect of specific RNA modification gene losses on the spectrum of modifications in some tRNAs. So we purify the tRNAs, fragment them with ribonuclease T1, put the tags on, iTrack tags, mix them together, do the mass spec. At the same time, we took the fragmented tRNA and we did a mass spec analysis, the heat map on the right, showing the relative amounts of a bunch of different modifications. This is our benchmark to compare the sequencing data. Now, we were able to get about 30% complete coverage of the 300 expected ribonuclease T1 fragments. We detected 100 in this particular case. Now we're using positive ion mode, which is not the most sensitive. And if we're switching now to negative ion mode, which ought to dramatically improve the sensitivity and probably also greatly improve the coverage here. So we're off to a good start. This is a place to improve in the future. All right, so let's look at one isoacceptor. This is tRNA cysteine GCA. And here we're ribonuclease T1 digestion gives us 4 fragments that are color-coded here, and we're going to quantify those 4 fragments. If we look at 3 of them in 6 different mutants, and these are different tRNA-modifying enzyme mutants in Pseudomonas, you can see that in amongst across that spectrum of mutants, this isoacceptor is pretty constant in the abundance. That is, these 3 fragments, which don't have any changing modifications, all come in pretty close to the same across all the analyses. So none of the mutations really affect the abundance of this tRNA isoacceptor in total. But now if we start to look at the anticodon loop fragment, there's some really interesting biology that pops up here. Now, in this particular isoacceptor at position 37, we can have any of 4 different chemical modifications with isopentenyl adenosine, I6A, and its derivatives, MS2I6A and MS2I06A. So let's have a look at this now and start to dissect this, that just this one isoacceptor in the population that's in these in Pseudomonas. If we look at the MIAA mutation, we lose all fragments containing I6A. It makes total sense for everybody in the audience because you know that MIAA is responsible for forming I6A. Knock it out, you've got no I6A and none of its derivatives. And so that's just sort of a proof of concept of where we're going. All right, now let's look at other fragments here. In the red box at the top, we're going to look at the fragment that's got I6A and HO5U in it. That's the black bar. Surprisingly, not surprisingly, I should say, when we knock out MIAB, that specific fragment increases significantly. Why? MIAB converts I6A to MS2 I6A. So if we no longer have the enzyme, we're going to get a backup of I6A. What's interesting is that it's always the I6A that's got HO5U. We'll get to that in a minute. If we look at the other mutants, the tRNA modifying mutants, there's not a big deal change going on. So those mutations don't significantly affect the level of the tRNA that's got this I6A HO5U in it. But now when we look at the fragment that's just got I6A, no HO5U, it's not changing much amongst the mutations, even the MIAB. So does this say something about the requirement of I6A before you put HO5U in? Well, we're going to work that out now using genetics and molecular tools to figure out what's the dependency for subsequent modification based on this analysis. Okay, again, when you knock out MIAB, not surprisingly, MS2I6A and MS2I06A both decrease in that mutant. Surprisingly, these specific modifications decrease in other mutants as well. And we're not sure why. We don't know if the mutations cause stress that reprograms the tRNAs and reprograms this specific population, but there is a decrease in these two modifications and some of these other mutations in that one isoacceptor. Okay, so now let's kick it up a notch. Let's take the pool of tRNAs and put a stress in there and look to see how the stress affects the population of fragments and modifications. Here we're looking at starvation, nutrient deprivation in Pseudomonas. We've got the wild type cells here, and we're looking at 3 different isoacceptors. Here's our cysteine GCA that we were looking at earlier. What we know about starvation in a lot of bacteria is that reactive oxygen species skyrocket when the cells get stressed like this. And there's an interesting feature of MIAB in that it's an iron sulfur cluster protein. If you have reactive oxygen species around, it's going to destroy the iron sulfur cluster and knock out the activity of MIAB. So when we have ROS, not surprisingly, MS2I06A drops significantly in all the isoacceptors that would have it. And we see that I6A goes up. Again, sort of a substrate product relationship. The enzyme backs up and we have an increase in I6A. Surprisingly, we also have an increase in I06A, which is made by MIAE. So now we don't know, did MIAE increase in abundance in response to the stress? Or did we just have more substrate that we now have the same amount of enzyme and it's just making I6A into I06A? All of this is to be worked out, but it reveals a point here that there's some novel signaling mechanisms that link these RNA modifications to different cell stresses in order to affect the phenotype and to adapt. Okay, so here we are. It's a multi-step process. We've done some optimizations. We got a lot more room to go in terms of things like fragmentation to get better coverage. Shenglong has got broad coverage because of the alkaline fragmentation that he uses. We're going to try some of these sequence-specific ribonucleases. We've got better tags. And we're now working on some capillary electrophoresis to get single nucleotide resolution of RNA fragments, which will go a long way toward improving resolution of the analysis, and then some new software that we're developing with Pat Limbach to analyze all of these fragments. So what's the goal here? The goal is to get to a point where a lot of people have talked about. How can we do high throughput analysis here? We've got our 96 well, 384 well plate-based system set up. Now we can do the modification analysis. Here we can do LC-MS RNA sequencing. We've just developed a new next-gen sequencing protocol that does not require gel purification to get rid of the primer dimers, and now you can fully automate next-gen sequencing library preparation to be able to take one RNA sample, break it up into groups, and go do multi-omic analysis on here for hundreds to thousands of samples. Okay, that's it. I have to thank the fantastic people who did all the work while I had jet lag and drank coffee, some fabulous alums that are out burning down the world with new companies and things, and some great collaborators. Thanks very much for listening.
Thank you, Peter, for your time and excellent talk. Really, really wonderful work. Thank you so much. I think we are scheduled right on time. That was a 12:10 stop Eastern Standard Time. And we're supposed to be back in 10 minutes for a panelist discussion, kicking off with Dr. Keach and from University of Utah. So this is, I think we should take advantage of the break. There's no questions right now in the chat. So let's save this for the panelist discussion. Thank you so much for coming back then.